Diane Gosselin, Rebecca Kush, and Guillaume Beslon to the stage. Diane and Rebecca are going to present a complementary view from two very different perspectives. Now, Rebecca is part of a nonprofit organization that saw the need to harmonize clinical data and the way that you measure disease and within a clinical trial. And Diane works for the Quebec CQDM, and it's a funding agency for innovation within your region or all of Canada?
Across Canada.
Across Canada. So she funds a number of projects. Not all of them are healthcare, but she has actually put some funding into Michelle's project. And
I'm very proud to do so.
Yeah. So I leave you two ladies to.
We'd like to show you to start off this discussion, and it goes back to some of what Ashley was talking about with respect to the feedback loop. So if we could show that slide, what we're doing is taking healthcare data and using it for research, and then the feedback loop from research to go back into clinical decisions is actually reported to take approximately 17 years. So I'm not even sure we're walking yet, but it's unacceptable. And in addition to this, the research studies are getting more and more complicated. They are generating more data, they're starting to use genomic data, and they're growing. And so Diane's going to talk next a little bit about the widening innovation gap that we're seeing.
Yeah, so I agree with you, Becky. I'm not sure we're walking yet. Especially at CQDM, we're concerned about the lack of productivity of the whole discovery process. So it's becoming more and more difficult to put some new drugs on the market, even though we are investing more and more in research. We are not able to come up with some drugs, more drugs approved every year. So CQDM is very concerned about that. It was created with that in mind, so we are trying, our mission is to fund tools that can accelerate the discovery process. So anything that can really facilitate the whole process. CQDM was created initially by three big pharma companies, Merck, Pfizer, and AstraZeneca, together with the Canadian and the Quebec government. And now we are very privileged to have 9 big pharma companies around CQDM. Again, the concept, well, probably you're more familiar with IMIs. CQDM is similar to IMI, and we're very privileged to have the good advices of Michelle Goldman on our board. So CQDM is smaller than IMI. We're focused in Canada, across Canada, and we have a very unique collaborative model where all the big pharma companies are sharing the cost of the research. So it's very important for us because it allows to create a financial leverage up to 25-fold. So because we have that financial leverage, we can afford to work on early stage research, we can de-risk research, and certainly we can work on research that a single pharma organization wouldn't be able to work on. So, yeah, so very important for CQDM from our perspective. If we want to go from big data to smart data, we need to work into a really collaborative way. And we do that at CQDM in many different ways. So first of all, as I mentioned, different big pharma companies who are initially competitors are working together, sharing the cost and the results of the research, but also making sure that academic researchers are going to work with private organizations, biotech companies, and we are funding these public-private partnerships. Now, that's not all. We need to make sure that the pharma industry is going to work in collaboration with these partnerships. So we have a mentorship program where we have mentors from the big pharma companies who are really closely working with our researchers to make sure that our technology is really aligned with the needs of the pharma industry. So what we mean by collaboration is also we need to work into multidiscipline if we want to bring big data from different scale to come up with something that we can use really and that is really aligned with the needs that we're pursuing. Next slide, please. So in CQDM, I don't have time to go too much into details, but we have a portfolio of 35 research projects. So I'm very proud to find Professor Bergeron, who is funded by CQDM, and you certainly did a lot of very nice progress with CQDM funds. But I would like to highlight some of the needs that we have in Alzheimer's disease, because this is a very good example where you have a lot of needs and not too many solutions that are coming up. Alzheimer's disease is a very complex disease, and we at CQDM, we have different ways to approach the problem. So one example is the project that on different animal models for Alzheimer's disease. So as you know, we have already on the market a lot of different models. They are not very predictive of what's going on in human. So with this project at BioSpective, which is a CRO based in Montreal, together with Pfizer and Merck, what we are developing is an imaging tool that allow, first of all, to characterize in depth the different models, models that are being developed by Merck and Pfizer. And so it's really nice, but we need to make sure that we have some ways to compare the results that we are generating. So with that specific tool, we are able to quantify the evolution of different biomarkers in time and compare, you know, within different animals and within different models. So at the end, we can come up, we're probably going to end up with different models that we can use together to see the evolution or the efficacy of a treatment. So that's one example, a multidisciplinary approach and a very nice interaction between the pharma industry and a biotech company. Now, the second example is about more clinical data. So, of course, in Alzheimer's disease, what we need is early diagnosis, first of all, and we need also to be able to predict response to treatment. So with this project led by Richard Hogg at University of Montreal, we are looking at different aspects of how can we bring different biomarkers into one single diagnosis. In this project, we are looking at the MRI results that we are combining with already existing biomarkers and clinical data to make sure that we're going to come up with something extremely predictive. Now, I don't want to go too much into the details, and I want Becky to maybe show us another aspect of smart data.
Sure, thank you, Diane. So my organization is called CDIS, Clinical Data Interchange Standards Consortium, which is quite a mouthful, but we started in 1997 as a totally volunteer group and incorporated as a nonprofit in 2000. And we now have 364, I believe it is, organizational members from all different types, academia, pharma, technology providers, and CROs make up the bulk of them. And we started by trying to figure out how to create data standards that would allow us to exchange data better. And we now have a suite of data standards. We initially started with trying to provide data to the Food and Drug Administration because all of their data was coming in paper, and their organization was becoming what was called a fire hazard, and they needed to get electronic data into their shop, and they wanted it in a standard format that would provide a way for them to use standardized tools to do the analyses. That is the back end of the process. Then we started also with a number of standards that will help generate that data and collect that data in a standard format. And I'll cut to the chase and say if the standards are used upstream, they're of the most value, and they add quality to the data as well as decreasing time and cost to the projects. So we say that standards bring order to complexity, and CDISC is actually becoming a global language for clinical research. So I'm going to give you an example because we don't use any data and we don't generate data. We're just trying to enable the exchange of that data. And one of our partners, the Critical Path Institute, started a coalition against major diseases, and they decided to begin with Alzheimer's disease. So I'm picking up the thread from Diane and saying, what are we doing to enable us to find treatments for Alzheimer's disease? And the Critical Path Institute approached a number of pharmaceutical companies who had failed trials, and their trials were done with a couple of hundred patients, and they said, what would happen if we could aggregate all these data into one database and start looking at a bigger amount of data? But they ran into this problem that you see on the slide here. Every one of them was collecting the data in a different way, in a different format. And one of the very standard tools, one of the questionnaires to assess cognition in Alzheimer's patients, was being used by each company in a different format. They were collecting the data in different sequences, and the data could not be aggregated. So they said, we need a standard, and we need to aggregate these data and map it into that standard. And so that's what they did, and they chose CDISC, and they got most of the data from the studies into the CDISC standard, but then the Alzheimer's-specific data is where we collaborated with them and came up with a new standard. So they were then able to aggregate initially 4,000 patients worth of data. It's now up to 6,000. The database is open. It's open and available for researchers to use, and they started being able to do some progression modeling that you see up here. And this is a quote from one of the people at CPath about how using the standards helped them get to the insights in a quicker way. So this is just one example in the field, again, of Alzheimer's, of where standards can help. And I'll close my presentation by just saying we now have a suite of standards that work for all clinical trials, and they're being used globally to go from protocol through data collection, through the analysis, and the reporting. And these standards are common across clinical trials like demographics and adverse event reporting, and we're now starting to add the disease-specific standards to that through a group we call Coalition for Accelerating Standards and Therapies, and it's a partnership with the Critical Path Institute. We also have some standards that help us bridge back into healthcare. So that's what we're doing to try to accelerate that cycle that we showed you at the beginning of this presentation.
Well, I invite you to have a seat. Now, I have a question for each of you. Come have a seat. The collaborative approach that is so important to how difficult is that to orchestrate within the pharmaceutical community? When you say you have 3 companies that are kind of, you know,
we have 9 now. 9. Yes. So I guess that, first of all, we need to make sure that we create a climate of confidence, because, as I mentioned, these are competitors. So we need to create an environment where we don't want to compete. We see the value of working together. And then, of course, with these tools, pharma companies are going to use these tools and then eventually compete with using the same tool but on different molecules. So the point is to really create a climate where we can create benefit for different pharma companies, first of all, but certainly also, and very importantly, to create benefits for all the stakeholders. So academic institution, private organization, and the pharma industry as well. So that's the point here. We need to probably understand each side and make sure that what we are going to do is going to collectively create benefits.
And Rebecca, we were talking before about this wonderful Alzheimer's unification way of presenting the clinical data. are pharma companies embracing this? Are they using it optimally, or, you know?
So we've had these standards being generated over the past 15 years, and a number of the pharmaceutical companies are participating in their use. And they use them in varying ways. And we just did an analysis of adoption with the Tufts Center for Drug Development. And unfortunately, there are still too many companies using them at the very, what I call the back end, where they collect all the data and then they map it into that standard. And that's a problem because it takes extra time, it takes extra money, and you can never get the data to quite fit into the standard. So we keep proposing, and so do people at FDA, including their statisticians, collect the data in that format in the beginning, and it'll flow downstream, and it's been shown to actually, you can start up a study in 70 to 90% less time when you use the standards in the startup stage of the study. So the Tufts study showed that about 90% of the pharmas are using the data at the back end and 20 to 30 at the front end, 20 to 30%.
Interesting. Okay, so
they are now going to be required to submit in that format, so I keep proposing that they try to save money and start by at least collecting the initial core data with a standard format.
Which, it's, I mean, given, having done some work in medical affairs, we spend a lot of time, even at its inception of a drug, you know, scripting key messaging and how you're going to discuss it. I mean, I would think the idea of how you're going to measure it and how you're going to compare it to what other companies are doing would also be something that you would, you know, be in the discussion.
And that's starting, and that's actually where the EMA is, the European Medicines Agency, because they want to increase transparency. So they are requiring now that trials be registered when they start and that data be shared at the end. And a lot of that's being shared in summary results, and so you can use some of the standard analysis data sets to show those summary data.
Well, I think you emphasize that we still have some hills to climb to make data smart. So Guillaume Beslon works for INRA, and he is giving us a computational biologist's perspective on big data. And he has some interesting views on the obstacles as well as ways to augment turning big data into smart data.
Okay, thank you, Rebecca. Barbara, sorry. So yeah, I'm not clearly from big data fields. I'm more a computational biologist. And, but I perfectly agree with our first speaker, Ashley, in that in big data we are in the infancy of the field. And we are able to walk or to make a step. Maybe not to walk, actually. We are not able to run. We want to run. And if we want to run, we have to be very careful not to run in the wrong directions. And in my talk, I will try to put the focus on, for me, the three main mistakes we may do if we run in big data. And the first one is actually, if it works, this one. Very classical, volume, velocity, variety, volume, velocity, variety, etc. And this is the first big mistake, focus too much on the data. If we want to progress in big data, we need to forget first the volume, the variety, and all these characteristics of our data, because this is not the point in healthcare. This is a question, this is a problem for computer scientists. This is not a problem for us. And actually, if you look at the data we have now in these domains, in the domain of healthcare, they are not so big. If you look at what the data we have in computer science, sorry, it's a little bit, okay, this one, for instance, well, many tweets, many pages on Facebook, etc. The volumes in healthcare are smaller than these ones. So this is not the point. So what is the point if we consider big data in healthcare? The point is that we have to keep in mind that if we use data in healthcare, then we are immediately in an interdisciplinary debate. Okay? And we need immediately to cross disciplines, to melt computer scientists, statisticians, mathematicians, biologists, of course, physicians, physicists, all these topics must talk one with the others if we want to work with big data. And if we have to talk in an interdisciplinary way of big data, then the definition of volume velocity is useless. The true definition of big data, at least for me, if we are in an interdisciplinary perspective, is simply data for which we don't have the methods on the shelves. But the method to do what? And this is the main question. If we want to start running with big data, we have first to understand precisely what we want to do with our data. It's easy to collect data. It's difficult to collect them in a standard way. I perfectly agree with that. But it's more difficult to standardize data than to collect them. So what we have to do first is having a purpose. And of course, the main purpose is to take care of the patient, to cure them, to prevent them to develop diseases. I don't know, many of these purposes. But these are not purposes we can express in terms of data. So we have to link the purpose with the data, and then we will be able to develop the field. This is, I think, the first mistake. I would say, don't listen to computer scientists. Sorry for the next two speakers. Or don't listen only to computer scientists. The second mistake, according to me, is this one. We have this naive view, or some of us, or some have this naive view of big data being what I call the patientomics. Okay, we will gather many data on the patients, and then we will be able to cure the patient. Okay, this is naive. Why? But why is this naive? Because data will never give you a way to cure the patient if you have the data on the patient alone. What you need is that you need some regularities in the data to be able to decide something from the data. And if you have this simple scheme, where are the regularities? They have to come in this scheme from the outside. And the main way to have them, of course, is to introduce in this scheme cohorts of patients. And with these cohorts of patients, we will be able to find regularities. But this has strong consequences. In that, if you look at the bottom of my slide, you cannot do personalized medicine anymore. Because what you will do is simply find other patients in your database that have the same disease, the same symptoms, similar symptoms, and try to cure your patient in the same way. But what if you don't have the patient in your database? Or what if you have the patient with an old data format? What if you have the same patient but with an old way of acquiring your data? You have to keep in mind that the way we acquire data evolves faster than the way we are able to cure the patients. So this is clearly a big problem, standardization, because we need to standardize what I have at the bottom left of my slide, given the fact that part of these patients, for data on these patients have been acquired with different technologies, with different levels of errors, with different levels of precisions. How can we make, or how can we extract regularities from that? But what now if we want to go back to personalized medicine? We not only need cohorts of patients, we need other things. We need to understand what occurs in this patient. We need to understand the disease. We need to understand the patient. And so actually what we need, and what we need for big data, is not only data. It's something more important, at least to me. It's science. What we need is to be able to understand the disease, to understand the patient, again, and then to be able to see, okay, what is the consequences of the data I collect on the patient at the level of understanding, not at the level of deciding. And then with this scientific knowledge, that of course you can extract from big data, then we will be able to go further and to go better in the direction of personalized medicine. So of course it doesn't mean that big data is useless. big data is extremely useful, but it is useful if you use it to develop knowledge first and then to apply knowledge in a second step. Now, my last point, and probably the last mistake according to me, is what kind of science we can do with big data. And there is a big risk there. The big risk is actually the, quite slow, yeah. So the big risk is that computer science, okay, is slow. The big risk is this one. What do I want to express on this slide? Big data focus on molecular level, mainly actually. And this is a risk because we change the way we are looking to the organisms. And if we collect many data at the molecular level, we don't collect data on the whole organism. We are not able to understand the whole organism at the molecular level. And this is something very important, and this is a big risk of big data, changing not only the way we look at the patient, but the way we try to understand the patient, and making us running in a, for me, clearly wrong direction. The direction of only molecular view on the patients, on the organisms, on bacteria, for instance. So what we have to do is we have to collect data, but not only at the molecular level. At all the levels of organization of a living being, be it the patients, be it the pathogens, I don't know. But globally, and I took this slide from Evelyn Fox-Keller, which is a philosopher I really like, living beings looks like this slide more than the previous one. We can discuss a lot on why they look like this slide, why they look like this figure. But for me, the reason is that it's because they have not been engineered. They have evolved. And evolution creates things simply by trials and error. So if, I don't know, the shoe here has a good action on the human, on the organism, then it will select the shoe or any other part of this system. And we have to understand this biological system at this level, at the global level and at the molecular level. I don't want to say that molecular level is useless. Again, molecular level and molecular biology is extremely useful, but it will be dangerous if it makes us forget the other levels of organization. And big data today have all the tools to collect lots of data, but only at the molecular level. It's much more difficult to collect data at the other levels, or it's much more difficult to call them big data. So just forget big data. Big data orients us toward molecules. Just speak of data. And data will cover all the range of organization of molecular beings. Thank you.
Well, thank you. So I'm relieved that big data is not the death of personalized medicine. But also as a physiologist, I think I would like to emphasize and maybe ask any of the panelists to comment on being able to take that broader view, that more holistic view of the person that is being treated and how healthcare is being practiced. Anyone care to comment?
Yeah, I agree entirely that it is important to get many different levels of data analysis. One of the really key things is identifying complex phenotypes that are themselves enormously integrative. And a really terrific one is heartbeat variability, because what that assesses for you, and what I mean by that is your heartbeats are irregular if you measure them very, very carefully. A lot of variability in heartbeat is very, very good. Very little variability is very bad. And what heartbeat variability does is synthesize and integrate what the parasympathetic system is doing as opposed to the sympathetic system. So you're integrating together at a very high level, very complex systems. And my argument has always been we'd like to have 50 complex phenotypes like that that could take us not just at the level of clinical chemistries or metabolites in the blood, but could really be assessing the systems themselves and how they operate in the organism. And I'm sure as time goes on, we'll come to understand many of these complex aspects of phenotype. And others do exist now, and it's going to be very important to interrogate them.
Thank you. Thank you very much.